Dr. Elinor Karlsson has always been curious about how living things work. She used to spend hours watching her family pets and wondering why animals and people are the way they are. In high school biology, she finally started to get answers to those whys, like why some people and animals get certain diseases while others don't. She went on to study biochemistry at Rice University and earned her PhD in bioinformatics from Boston University. She started her own research group at UMass Medical School in 2014. Back then, most genetic research focused on humans, which was expensive and required a lot of fancy tech, so she turned to dogs since they share many of the same diseases and traits as humans, but have simpler genetic patterns. Outside the lab, Dr. Karlsson loves hiking and spending time with her dogs. She's also big on science communication, which is breaking down complex genetics topics so anyone can understand them. She leads Darwin's Dogs, a citizen science project where dog owners can help study how genetics influence dog behavior in diseases like OCD and cancer. She also co-leads Zoonomia, a global project comparing the genomes of more than 240 mammal species to find the most important parts of DNA. I really admire how she not only studies animals to help them scientifically, but also clearly loves and cares for them personally. Dr. Karlsson's field is comparative genomics, which basically is comparing DNA across different species. Her main research questions are: how do genes influence behavior and disease, and which parts of the mammalian genome are so important that evolution has kept the same across species? One recent primary research article that I read of hers is called Comparative Genomics Reveals Shared Mutational Landscape in Canine Hemangiosarcoma and Human Angiosarcoma. And figure 3 in this asks what mutational process are driving the genetic change in canine hemangiosarcoma tumors. And researchers basically wanted to see if the mutation came from normal aging, environmental exposure, or other biological mechanisms, and if those patterns matched human angiosarcoma. To do this, researchers used whole exome sequencing, which sequenced the coding regions of DNA from dog tumors, and comparative analysis, which compared the discovered dog signatures to known cosmic human cancer signatures. And the experiment was basically conducted where a tumor and normal tissue samples from dogs with hemangiosarcoma were sequenced, and the team identified somatic mutations and analyzed the patterns of nucleotide changes to infer the mutational processes involved. They matched normal DNA from each dog to separate tumor-specific mutations from inherited variants, to be used as a control. And overall, my main conclusion from the figure is that the dominant mutation null signature matched cosmic SBS1, which is associated with normal aging and cell division, which means most mutations likely result from natural biological processes and not environmental damage. The similarity between dog and human tumors supports using dogs as a comparative cancer model. Karlsson's group discovered that dogs and humans share key cancer driver genes, such as TP53 and PI3K pathway genes. Dog behavior and disease are not determined solely by breed, as shown through citizen science pet genomics, and through Zoonomia, her lab identified highly conserved DNA regions that help pinpoint genes critical for health and development across mammals. The next steps in her research include studying larger canine cancer cohorts to uncover rare genetic drivers, standardized genomic analysis pipelines across species, and to expand Darwin's arc to include more animals and traits. These are my citations. Thank you so much for watching.